Assalamu alaikum and welcome everyone to our molecular biology ASCP preparation series. This program is all about providing real-life MCQs for your certification success. The MCQs we'll discuss today and in upcoming season have been carefully collected from several years of past exams to give you the best preparation possible. I'm Dr. Lai Abdelati, certified as international technologist in molecular biology, and I'm really excited to share my knowledge and experiences with all of you through this series. I also encourage everyone taking the test to share your question with us so that everyone can benefit. All right? So let's dive in together, but don't forget to subscribe to our channel to receive all the latest lecture. Are you ready? Let's get started. The molecular science part is divided into four sections: nucleic acid chemistry, basic molecular theory, biochemical reagents, and genetics, which together make up 20 to 25% of the total molecular ASCP exams. In our previous lectures, we covered multiple choice questions related to the nucleic acid chemistry. We then moved on the basic molecular theory sections and completed the replication part. Now, we'll delve into the transcription section, alright? For those who missed the previous lectures, I have added the links to those lectures in the bio below for your reference, okay? Now, let's focus on the question related to transcription in eukaryotic cells and review ASCP questions from the previous years, alright? Alright, the first question we have today is which RNA polymerase is responsible for synthesizing messenger RNA in eukaryotic cells? The correct answer is RNA polymerase II. RNA polymerase II is essential for the production of messenger RNA in eukaryotic cells. All right, let's move on. The next question is, what is the function of the TATA box in transcription initiation? First, we have to know what is the TATA box. TATA box is a conserved sequence found in the promoters of eukaryotic genes. All right, it binds transcription factor and facilitate the accurate positioning of RNA polymerase II at the transcription start site. So the correct answer here is what is the function of TATA box is directs RNA polymerase to the transcription start site. All right, let's move on. All right, the question here is which transcription factor is the first to bind to the promoter during the initiation of transcription? The correct answer is transcription factor 2D. Transcription factor 2D, which include the TATA binding protein, is the first transcription factor to bind to the TATA box in the promoter. All right? It acts as the scaffold for the assembly of other transcription factors and RNA polymerase II. Okay, let's move on. Okay, the question here, what is the role of the 5' cap added to eukaryotic pre-messenger RNA? Actually, the 5' gap is a modified guanine nucleotide, protect the pre-messenger RNA from degradation by X nuclease and help the ribosome recognize the messenger RNA for translation. All right. So the correct answer here is protect messenger RNA from degradation and assist in translation initiation. Okay. Let's move on. Okay. The next question is which enzyme is responsible for adding the poly A tail to eukaryotic pre-messenger RNA? All right, the correct answer is poly A polymerase. Poly A polymerase adds a poly A tail to the 3' end of pre-messenger RNA after cleavage. This tail enhances messenger RNA stability and assists in nuclear export and translation. Okay, let's move on. Okay, the next question is in eukaryotic transcription, what is the role of transcription factor 2H? Actually, we have to know transcription factor 2H has helicase activity to unwind DNA at transcription start site and kinase activity to phosphorylate the C-terminal domain of RNA polymerase II, enabling transcription elongation. All right. So the correct answer here is unwinds DNA and phosphorylate RNA polymerase II. All right, let's move on. Okay, the next question is which of the following processes is unique to eukaryotic transcription compared to prokaryotic transcription? Actually, the addition of 5' cap and poly A tail are specific to eukaryotic transcription and are a part of RNA processing, which is absent in prokaryotes. So the correct answer here is addition of 5' cap and poly A tail to pre-messenger RNA. Let's move on. The question here is what is the function of enhancers in eukaryotic transcription? Actually, enhancers are DNA sequence that enhance the transcription of specific genes by binding activator proteins, which then interact with the transcription machinery as the promoter. All right. So the correct answer here is function of enhancer in eukaryotic transcription is increase the transcription of specific genes by interacting with the transcription factors. All right. Okay, the question here, which modification must occur for RNA polymerase II to transition from initiation to elongation? The correct answer is phosphorylation of its C-terminal domain. Actually, the phosphorylation of RNA polymerase II C-terminal domain by transcription factor H allows the polymerase to escape the promoter and transition into elongation phase. All right, let's move on. The next question, what is the spliceosome composed of? Actually, spliceosome is a complex of small nuclear RNAs such as U1, U2, U4, U5, and U6, and associated proteins. It removes intron from pre-messenger RNA and joins exon together to form mature messenger RNA. Okay, let's move on. Let's see the next question. What marks the end of transcription in eukaryotes? All right. Transcription in eukaryotes ends when pre-messenger RNA is cleaved at a specific sequence downstream of the coding region and poly-A tail is added to the transcript. Okay, so the correct answer here is cleavage of pre-messenger RNA and addition of poly-A tail. Okay, let's move on. Which component is responsible for recognizing and binding to the promoter region during transcription initiation in eukaryotes? Which one? All right, the correct answer is C. TATA binding protein, a part of what? A part of the transcription factor 2D complex. Okay, specifically recognize and binds to data box where in the promoter region. This is the first step in formation of transcription pre-initiation complex. All right, let's move to the next question. The next question is, which eukaryotic RNA polymerase transcripts transfer RNA genes? Okay, the correct answer is RNA polymerase III. RNA polymerase III transcripts small RNAs including transfer RNA and 5S ribosomal RNA, which are necessary for what? Which are necessary for protein synthesis. It's specific to non-coding RNA transcription. Okay, let's move on. Okay, the question here. What ensures the proper export of mature messenger RNA from nucleus to the cytoplasm? Okay, the correct answer is nuclear export signals and binding of specific proteins like the exon junction complex. I will explain it in a simple way. Actually, after splicing process occurred, specific proteins such as the exon junction complex bind to messenger RNA, facilitating its recognition and transport through the nuclear pores to the cytoplasm. Right? So the correct answer is D. Let's move on. Next question. Which sequence in eukaryotic messenger RNA is critical for translation initiation in cytoplasm? The correct answer is Cossack sequence. Actually, the Cossack sequence helps ribosomes recognize the start codon. The start codon is AUG, alright, in eukaryotic messenger RNA. Ensures efficient binding of the ribosome for accurate translation initiation. Okay, so the correct answer is Cossack sequence. Let's read the next question. What is the purpose of mediator complex in transcription? The correct answer is connects transcription factors and RNA polymerase II to coordinate transcription activation. Okay, actually, the mediator complex bridge transcription factors and RNA polymerase II. So this enabling the efficient assembly of transcription machinery and activation of transcription. Okay, let's move on. Okay, the question is, which sequence signals the end of transcription in eukaryotic cells? All right, the correct answer is polyadenylation signals. Actually, the polyadenylation signals marks the site where the RNA transcript is cleaved and poly-A tail is added to terminate transcription. Okay, let's move on. All right, which histone modification is typically associated with the transcriptionally active chromatin? All right, let's see the correct answer. All right, the correct answer is histone acetylation. Histone acetylation neutralizes the positive charge of histones, reducing their affinity for negatively charged DNA, thereby opening chromatin and facilitating transcription process. Okay, let's move on. The next question is, how does chromatin remodeling affect eukaryotic transcription? The correct answer is alters the accessibility of DNA to transcription factors and RNA polymerase. Actually, chromatin remodeling uses ATP-dependent complexes to repositions or remove nucleosomes. Okay, making specific DNA regions more accessible for what? For transcription process. Okay, let's move on. Okay, let's see the next question. What happens during the elongation phase of eukaryotic transcription? The correct answer is RNA polymerase II sensitizes RNA complementary to the DNA template strand. Okay, during elongation, RNA polymerase II moves along the DNA template strand, right? Sensitizing a complementary RNA strand by what? By adding nucleotides. Okay, let's move on to the next question. All right, the next question is, what prevents premature degradation of eukaryotic messenger RNA during transcription? So the correct answer is addition of 5' cap and 3' poly A tail. All right, the 5' cap actually and 3' poly A tail protect the pre-messenger RNA from enzymatic degradation such as nucleases. Okay, and help facilitate ribosome attachment during translation process. Okay, let's move on. The question here is, which regularity element can enhance transcription despite being located far away from the gene in controls? All right, the correct answer is enhancer. Actually, enhancers are regulatory DNA sequence that can increase transcription levels of target genes regardless of their distance or orientation relative to the promoter. Okay, so the correct answer is enhancer. All right, let's see the next question. Which phase of transcription is regulated by the phosphorylation of the RNA polymerase II carboxyl terminal domain? All right, the correct answer is transition from initiation to elongation step. Okay, actually, phosphorylation, as I said earlier, phosphorylation of carboxyl terminal domain of RNA polymerase II by what? By transcription factor H is the critical step that transition RNA polymerase II from what? From initiation to elongation step. Okay, let's move on. Okay, here you see the same the same previous question but in different way. The question is, what is the purpose of the carboxyl terminal domain of RNA polymerase II? Okay, let's see the correct answer. Okay, the correct answer is regulating transcription and RNA processing. As I said, the carboxyl terminal domain of RNA polymerase II is phosphorylated during transcription process, regulating the transition from what? From initiation to elongation, and recruiting factors involved in capping, splicing, and polyadenylation. Okay, I hope it's clear for you. Okay, let's move on. Okay, the next question is, which RNA processing step removes introns from eukaryotic pre-messenger RNA? Okay, the correct answer is splicing. Actually, in splicing process, what happened? Splicing removes non-coding introns from pre-messenger RNA, leaving only exons to be joined together, forming mature messenger RNA for translation. Okay, let's move to the next question. The question is, which element is found in promoter region of most eukaryotic genes? Okay, let's move on and see the correct answer. Okay, the correct answer is TATA box. The TATA box is a conserved DNA sequence in the promoter region that help initiate transcription by serving as the binding site for transcription factors and RNA polymerase II. All right, let's move on. Okay, the next question is, what modification is added to the 5' end of eukaryotic messenger RNA during transcription? The correct answer is methylguanosine cap. The 5' methylguanosine cap is added to eukaryotic messenger RNA during transcription. It protects the messenger RNA from degradation and facilitates ribosome binding during translation. Okay, let's move to the next question. Okay, the next question is, what is the role of 3' poly A tail in eukaryotic messenger RNA? Okay, the correct answer is messenger RNA stability and nuclear export. Actually, the poly A tail, a string of adenine nucleotides, stabilizes messenger RNA molecules and help them exit the nucleus. It also prevent degradation in the cytoplasm. All right, let's move on. All right, which eukaryotic RNA polymerase sensitized ribosomal RNA? All right, the correct answer is RNA polymerase 1. RNA polymerase 1 transcribes ribosomal RNA, which form the structure and functional core of ribosomes and is essential for protein synthesis. All right, let's move on. Okay, the next question is which transcription factor binds directly to the TATA box? All right, the correct answer is TATA binding protein. Actually, the TATA binding protein, which is the part of transcription factor TF2D, has the important job in starting transcription. It is attached to the specific DNA sequence called TATA box. So, this attachment helps set up the pre-initiation complex, which is needed for to begin the transcription process. All right. So let's move on. All right, the next question is how is the fidelity of eukaryotic transcription ensured? All right, let's see the correct answer. All right, the correct answer is proofreading activity of RNA polymerase II. As I said earlier, RNA polymerase II has proofreading mechanism that ensure accurate transcription by correcting mismatched nucleotides. All right, let's move on. The question is what happens to eukaryotic pre-messenger RNA after transcription? Actually, eukaryotic pre-messenger RNA is proceed by splicing. Splicing it means remove of introns, right? Capping addition of 5' cap, and polyadenylation addition of 3' poly A tail, okay, to become mature messenger RNA ready for translation. So the correct answer here is B. Okay, let's move on. All right, here in the table we have overview of the key transcription factors which are vital protein involved in regulation gene expression in eukaryotic cells, right? These factors are crucial in determining when and how specific genes are activated or silenced, allowing cells to adapt to various signals and maintain their normal function. It is important to study and remember this information as some exam question will be derived from this material. Okay? Focus and focus and focus, guys. Alright, that's all for today. Please don't forget to like, comment if you have any question, share and subscribe to our channel to receive all the latest lecture. Salam.